All right, let's talk about something fascinating. We have these incredible tools like fMRI that let us peek inside the living human brain. But the data we get from them is, well, it's messy. So we have to clean it up. You'd think that if everyone used the same cleaning tool, we'd all end up with the same clean picture, right? Well, as we're about to find out, the puzzle is way more complicated than it looks. So just imagine this for a second. You take one set of brain scan data, you give it to 70 of the best neuroimaging labs on the planet, and you ask them all the same questions. You would absolutely expect them to come back with pretty similar answers, wouldn't you? I mean, it's the same data. Well, this wasn't just a thought experiment. It really happened. And the results, they were a real eye-opener. Now, to really get why this is such a big deal, we need to rewind a bit and look at how things used to be done in fMRI analysis. For the longest time, it was, for lack of a better term, the Wild West. Every lab was kind of on its own. And hey, don't just take my word for it. This quote from the creators of a tool we're going to talk about says it all. Ad hoc workflows. That's a nice scientific way of saying everybody was just making up their own way of doing things. Every lab had its own secret sauce, its own special process for cleaning up their brain imaging data. Think about it, like baking a cake. If you give 70 master chefs the exact same flour, sugar, and eggs, but they all use their own secret recipe, different oven temps, different mixing techniques, different spices, are you going to get 70 identical cakes? No way! And that right there is the core of the problem we've been facing in neuroscience. See, raw fMRI data is not a clean picture. It's super noisy. You've got noise from the person moving their head, from them breathing, even from the scanner itself. So to handle this, every lab built their own custom workflow. But that just led to chaos. If my lab found something and your lab didn't, was that a real scientific discovery, or did we just clean our data differently? It was a huge threat to being able to reproduce scientific findings. So to tame this Wild West, the field needed a new sheriff in town. It needed a standard, a protocol that everyone could use and trust. And that's where this amazing tool called fMRiprep comes into the story. It's really been a game changer. The goal behind fMRiprep was simple, but huge. Create one powerful, dependable pipeline to get fMRI data ready for analysis. The whole idea was to give every scientist the same high-quality, reproducible starting point, to finally end the era of secret recipes and give everyone some common ground to stand on. And the way it's built is just brilliant. First off, it's robust, meaning you can throw almost any kind of fMRI data at it, and it just works. It's also incredibly high quality because it doesn't reinvent the wheel. Instead, it takes the best of the best tools from all the major software packages and puts them together in a smart way. But maybe the most important part? It's transparent. It's not a black box where your data goes in and some magic answer comes out. It's a glass box that gives you these amazing visual reports so you can see exactly what happened to your data every step of the way. So, what does this glass box actually give you? Let's take a peek inside the output from fMRiprep, because this is where its real power to improve our understanding of the brain's anatomy really shines. Okay, for every single brain scan you run, fMRiprep creates this beautiful interactive web page, a report. And this isn't just a boring log file. It's a visual walkthrough of the whole process. You can see before and after pictures of all the key steps, so you can actually check with your own eyes that the software did a good job. That builds a ton of trust. So when all is said and done, fMRiprep hands you 3 key things. 1, the clean brain images, all set for your actual science. 2, those awesome visual reports for quality control. And 3, super detailed files that list all the noise it found and removed, so you can account for it in your statistics. Now this right here is one of the biggest leaps forward. For years, brain data was analyzed in what we call volumetric space. Think of it like building a model of the brain out of tiny little Lego blocks. The problem is, the brain isn't blocky. It's an incredibly folded sheet. So fMRiprep also gives you the data mapped onto a surface space. It's less like Legos and more like perfectly shrink-wrapping the data onto the brain's actual folded anatomy. The precision you get from this is just way, way better. It stops signals from different parts of the cortex from bleeding into each other just because they're close together in those folds. Okay, so we have this amazing, precise, transparent tool. Problem solved, right? Every lab can now start on the same page with perfectly prepared data. This is where the story gets really interesting, thanks to a huge study called NARPS. So let's ask that question one more time. Now that we have a standard tool like fMRIprep that gives everyone the same high-quality starting line, the problem of reproducibility is fixed, right? A study called NARBS decided to put that exact question to the test. And this wasn't a small thing. They got 70 independent teams of scientists from all over the world to analyze the exact same fMRI dataset. And what happened? Well, the results were all over the map. When asked if a hypothesis was supported by the data, sometimes half the teams would say yes, and the other half would say no. For 5 of the 9 hypotheses they tested, there was basically zero consensus. It was a scientific free-for-all. And I want to be clear, this wasn't just a little bit of disagreement, it was massive variability. On average, about 1 in 5 teams came to a different conclusion than the majority. It was about halfway between everyone agreeing perfectly and just pure random chance. How is that even possible? Well, here's the kicker. This one sentence from the paper says it all. No 2 teams chose identical workflows to analyze the data. Even though they all started with the same clean ingredients from fMRIprep, they all chose a different statistical recipe for the final analysis. And that discovery really points us to the next great challenge for the field of neuroscience. It turns out it's not just about how you clean the data, it's about what you do with it after it's clean. And let's be crystal clear, the problem wasn't the pre-processing. In fact, almost half the teams in the study used fMRIprep. The tool did its job perfectly. But whether a team used fMRIprep or their own custom method had basically no impact on the final variability. The problem started later in the process. So if you look at the entire workflow, you can see what happened. Step 1 is collecting the data. Step 2 is pre-processing, and fMRIprep put a giant checkmark next to that one, basically solving it. But all that chaos and variability, it just moved downstream to step 3, the statistical analysis. That's the new Wild West. There's a term for this, researcher degrees of freedom. It sounds fancy, but it just means all the thousands of little choices a scientist has to make when they build their statistical model. Things like, how do you handle an outlier? Which variables do you control for? Every single choice is justifiable, but when you add them all up, they create this huge universe of possible analysis paths. And that leaves us with this huge, important question for the future. Creating standard tools like fMRIprep was an absolutely critical first step. It solved one part of the puzzle, but in doing so, it revealed the next, maybe even bigger frontier. So how do we navigate this massive universe of choices that comes next? Figuring that out is the next great challenge for the science of brain imaging.